today we are lucky and we have invited Dimitris Xirodimas just to come here to Dival to know us. He came here because different reasons. We are not going to explain, but some familiar reasons also. And he's here. We are lucky, and he's in Cantabria just for 1 week. I told him just to come to visit us, to know our institute, and to explain us his line of research for so many years. His subject is protein aggregation. He has been collaborating with some of our investigators here, like Carlos Pipão, who is here, but also with some other collaborators close to Cantabria. And we are lucky having him. We couldn't do it in Wednesday as is usual, but I really thank people who comes here to see what he's doing. Dimitris is a researcher that started in Scotland. He was there for the PhD and also for a postdoc. Thereafter, you changed to France and you established your own line of research. He's the leader of a group in cell biology there and working on protein aggregation in Montpellier. Okay, he's very active, I tell you, and he's trying to explain us in a, let's say, easy way, and he's very basic researcher, but we were talking about this institute and how we try to do clinical translation from our research, and he's going to give us a view of his doing there in Montpellier. Dimitris, thank you very much. Thank you so much for coming here, for being available to give a talk and to, you see here, there is many young people, and I want you to motivate them.
Yes.
Working there. Okay, thank you so much.
Thank you. Well, thanks a lot for this very kind introduction and for the invitation here, and all to the Zoomers that they are participating in this talk. Thank you for coming. As Marco said, today I will try to present what we do in the lab. We are a fundamental research group. So I'm not going to bombard you a lot with mechanistic details and a lot of mechanistic data, but just to give you a bit of an overview of the concepts we try to develop in the team, fundamental concepts with clinical applications, because something now that we were very interested to exploit more, which we're not experts, but something that we want to exploit a bit more. And as Marco said, I mean, the key sort of aspect that we try to understand is that how we can exploit, how to understand the so-called protein quality control. In other words, how the system deals with the quality of proteins in cells, particularly with protein aggregation, understand the basic molecular basis of protein aggregation, and potentially how you can exploit this information for therapeutic intervention, both for neurodegeneration and cancer. I will give you a couple of examples today. Okay, so this is a very sort of basic concept of molecular biology, of how, you know, from our genome we can make proteins, and proteins is the workload of in the cell. That's the machine that is performing all the biological functions in the cells. But now we know that these basic elements in the cell that are constantly exposed to stress. So I guess you all know the so-called genotoxic stress. In other words, the stress conditions that induce both the DNA or RNA protein damage, but the damage that we're particularly interested in is what we call the proteotoxic stress. In other words, the conditions that induce, protein damage. And now we know that the system has developed, or the cells have developed, a very sophisticated elements of detecting, repairing, and eliminating both genotoxic and proteotoxic, induced damage. And this is very critical because in the absence of elimination, in the case of DNA, you all know that if you don't eliminate DNA damage, that may often leads to the development of diseases like cancer. But also, if you don't manage to eliminate damaged proteins, this is now known that is related to disease development. Most profoundly in neurodegeneration, that's very well established, and I'll give you examples today. But we also now understand that cancer can also be linked to the lack of elimination of protein damage. And in our lab, what we're trying to understand is that how this process is controlled by this family of what we call now ubiquitin and ubiquitin-like molecules, and most particularly the protein quality control system. So, So what is ubiquitin? What is ubiquitin-like molecule? So ubiquitin is belongs to a family of small proteins. So this is the structure over here. They're small proteins, they're extremely highly conserved. They're about 76 amino acids in length, about 8 to 9 kilodalton of size. And what they do is they're constantly modifying other proteins. So here we're talking about a post-translational modification, similar to what you guys know about phosphorylation, acetylation, and this type of post-translational modifications. Now, what is the way that these proteins work is, as I mentioned, they are post-translational modifying substrates. So they can do it in many different ways. They can modify proteins as a single moiety over here, or they have also the ability to make the so-called chains. In this case, you see the formation of ubiquitin chains. Because in this case, because the molecules that they modify the substrate is a protein, it can also modify itself. So this formation of ubiquitin chains, but also you can have the formation of chains with all the ubiquitin-like molecules, creates distinct molecular biological outcomes. The most known function of these ubiquitin chains is to target proteins for proteasomal degradation. So when proteins, they are targeted or they're modified by these ubiquitin chains, this creates a signal that's recognized by the proteasome in order to degrade the substrate. And that's the most well-known function for ubiquitin, but not the only one. But as I mentioned, ubiquitin is the founder member of a growing family of proteins, which we call now ubiquitin-like family of proteins. So now we know there's about 16 other small proteins belonging to this family. The most known, it's the SUMO ubiquitin-like molecule, and A8, that's the one that we sort of focusing on, and that's what also Carlos has done a lot of nice work in his laboratory. And here in this sort of cartoon, you see how, what is the mechanism by which these molecules are finally modifying the substrate proteins. So in order for these proteins to end up down here, modify the substrates, they need the activity of 3 enzymes, which we're now calling E1, E2, and E3 enzymes. So what's happening, these enzymes, they're passing, in this case, ubiquitin or the ubiquitin-like molecules from one to the other. And finally, the E3 enzyme, which we call it E3 ligase, it directly recognizes the substrate and is finally putting, in this case, ubiquitin on the substrate as a post-translational modifier. What is important here to know is that each pathway has its own unique set of enzymes, E1, E2s, and E3. So ubiquitin, A8, and SUMO have their own unique enzymes, and that provides specificity in the system. It provides specificity of how each of these ubiquitin-like molecules can, choose their, substrate at the end in order to elicit their biological function. Now, another complexity in this field is that apart from the addition of the formation of what I call the ubiquitin or homotypic chains, these molecules can crosstalk to each other. So in particular, when we expose cells to stress, and that's the thing that we are sort of interested in, these ubiquitin-like molecules can form the so-called hybrid chain. So in this case, you can have a ubiquitin chain that also A8 is incorporated, or SUMO is incorporated to ubiquitin chains, or between SUMO and A8. So here, just to illustrate how complex the whole system is, particularly upon stress conditions, particularly upon conditions where you're forming protein aggregates, when we're forming, protein misfolding. And the whole idea here is to understand how these molecules deal, or what is their role in terms of dealing with protein damage, in dealing with protein aggregation, in dealing with the accumulation of misfolding proteins. So here, again, I will highlight how these proteins are operating upon stress conditions. So native proteins, they're constantly exposed to a whole set of stress conditions, which we now define prototoxic stress, which includes heat shock, inhibition of the proteasome, exposure to chemical, chemotherapeutic, DNA damage, oxidative stress. All these conditions, they're causing proteins to losing their sort of wild type normal conformation, and they're getting unfolded. And the first line of defense is that chaperones, they are recognizing these misfolded proteins in order to get them back into their native state. But if this line of defense fails, this is where the ubiquitin system gets engaged. It recognizes these misfolded proteins in order to target them for proteasomal degradations, as we discussed. But if this line of defense fails, for example, if you have now misfolded proteins that they cannot be targeted, now the second line of defense is that the system accumulates these proteins in what we're calling now as protein condensates. So what are protein condensates? Protein condensates are inclusions that, many of these misfolded proteins, they start accumulating in these inclusions. And this happens because misfolded proteins, they're exposing hydrophobic areas that they don't like interactions with the liquid aqueous solution. They start sticking to each other and they're forming these reversible, condensates, which they are characterized by what we call now liquid-liquid phase separation. So in this cartoon, it just describes this process. So here we have proteins that are homogeneously dispersed and they're mixed in the sort of cytoplasm of the nucleus. But when you induce, for example, stress, the accumulation of misfolded proteins, it tends to causing the misfolded proteins to phase out from the rest of the environment, of the aqueous environment, and form this sort of a new phase that is now a condensate, an inclusion, and which most likely, or most of the cases, includes misfolded proteins, but also RNA. And that's sort of something very important, that these inclusions, they're not just proteins, but also contain RNA complexes, which they're becoming super important. And basically the concept here is that you can imagine as when you drop an oil into a water. So that's a sort of concept of phasing out of an aqueous solution, and forming this sort of condensate inclusions which they contain protein and RNA complexes. Now, this is sort of interesting. That is part of the physiological response. That's what happens every time you expose the cell to stress. But these condensates, they can also very often can lead to the formation of toxic irreversible protein aggregates. And this can be caused if you expose the cells to excessive stress or mutations that exist in proteins that are found in many diseases, mainly neurodegenerative diseases. So all these conditions, they can cause now the conversion of something that normally is protective, the formation of these inclusions, into something that is very toxic. And now what we know is that the ubiquity system now is managing to somehow recognize in either the condensates or even toxic aggregates, and in some cases it promotes their elimination. So this is what recapitulates, if you like, what we define as the protein quality control system, and how ubiquity in this case is involved in recognizing misfolded proteins, targeted for their degradation, or dealing with the formation of these inclusions that they are taking place either in normal conditions, stress conditions, or in pathological, toxic conditions. Now, another concept is that this process of formation of aggregates is very well compartmentalized. In other words, that the cell, and forget now the details in this busy scale, but what this slides over here shows is that the formation of many different types of aggregates or condensates that can take place in the cytoplasm or in the nucleus. And the system is very able to detect them at different compartments and deal with them. So at the moment, we know very well how the cytoplasm is dealing with these protein inclusions. The nucleus, though, is not very well understood. So, for example, how nuclear aggregates, they are formed, how they get eliminated, is something that is not very sort of understand. And this is something that we try to understand, and in particularly, if there is a specific pathway that operates in the nucleus to deal with this type of protein aggregates. Now, in terms of pathology, how these ubiquity-like proteins and aggregates are involved in pathology. Here, it's just examples of neurodegeneration, which is the best sort of pathology that is linked to protein aggregation. Here, you can see different type of neurodegenerative diseases, the formation of aggregates. And what is important, it's all these ubiquity-like molecules, they all accumulate in these aggregates. And still we don't understand how they accumulate there and why they accumulate there. What is the role of accumulation of these ubiquity-like molecules in all these, what we call now proteinopathies. Now, another big question in the field is that, okay, you have the formation of these aggregates in all these neurodegenerative diseases, but is it something protective? Is it something sort of beneficial or something related to the disease? For example, if we manage to eliminate these aggregates, we will be able to ameliorate this disease phenotype. In other words, can we exploit this aggregate formation for treatment for neurodegeneration? treatment. And evidence now recently come that this potentially is the case. In other words, that by eliminating these aggregates may be actually beneficial for treatment. And in this case, this is like very, brief sort of example that experiments devise, systems where they can basically force the ubiquitination of these aggregates. In this case, in this sort of Alzheimer's disease a model where it's characterized by the formation of these tau fibril aggregates, and by forcing the ubiquitination, they managed to force the elimination of these aggregates from this disease, and this was shown to be related to the amelioration of tau-related pathology. And this is important now for two different reasons. The first one is that it shows that indeed, by eliminating aggregates, is something beneficial for the new generation. But from a conceptual point of view, from a mechanistic point of view, it raises the question of if ubiquity-like molecules, they're already there in the aggregates, why does the system does not lead to their elimination? Why the endogenous system fails to detect this ubiquitin that is there in order to promote the aggregation, and why by putting more ubiquitin into these aggregates, that triggers the elimination by the endogenous system. So these are questions that are still sort of fundamental. We still don't know the answer to this, but at least what this experiment showed is that by eliminating the protein aggregates through this ubiquity system can be regarded as a potential beneficial approach. Now, as I mentioned, the interest of our group is on this ubiquity-like molecule, this what we call NEDate. And we now have evidence that potentially can be related to protein aggregation related to neurodegeneration. So far what I described to you how these ubiquity-like molecules, they are modifying substrate. In other words, I described you the forward reaction by the enzymes of E1, E2, E3. But these reactions, they're they're always reversible. In other words, the existing enzymes, which we call them deconjugating enzymes, and their role is to constantly remove the ubiquity-like molecules from NEDate in order to form like a cycle of conjugation and deconjugation. So this is an extremely dynamic element that exists, like if you like, in resemblance to what you know about phosphorylation, that you have the action of kinase and phosphatases, which they put on and they remove phosphate from protein substrates. So in relation to NEDate, there are two key NED-deleting enzymes. The one is called COP9, but the enzyme that we're interested in is called NEDP1. And this is what happens if you basically remove this enzyme from cells. So here's an experiment where we make a knockout of this enzyme, and you can see that you can see that the deletion of this enzyme causes a massive accumulation of NEDate conjugates in cells. And this is the reason, because by removing this enzyme, all NEDate now is trapped into this state. There's nothing to remove it. So you have this sort of dramatic accumulation of NEDate conjugates. And one of the key interests of the lab is to find the biological outcome of this accumulation. What does this really mean for the cell? So one of the things we found that potentially was interesting for us is that this enzyme is potentially related to this neurodegenerative disease, which is called amyotrophic lateral sclerosis. And I will tell you about this in a minute. So here we're looking at the activity of the enzyme in the brain of animals, either wild-type mice or mice that they suffer from this disease, NALS mouse model system. And we found that in the pathology, the activity of this enzyme is upregulated. So in other words, it does the opposite of what we see of the knockout. So for us was trying to understand what is this balance of the NEDate is implicating in this disease, and what is the function of this enzyme. So it turns out that the role of this enzyme in biology is to control a form of condensates that they induce upon oxidative stress, which we call them stress granules. So these stress granules, they are, as I mentioned, they are related to oxidative stress. So when you expose cells to oxidative stress, you see the formation of these inclusions in the cytoplasm of the cells, which is related to protein synthesis. In this case, what's happening, you put stress, proteins getting misfolded, protein synthesis is shuts down, and many of these misfolded proteins, they go into these inclusions. And as you can see, when the stress goes away, these inclusions now, they get eliminated. The system is dealing with this. Protein misfolded is going to degradation, and the system can sort of restart again. So this process is a reversible physiological stress response that happens to oxidative stress when you have the accumulation of misfolded proteins in the cytoplasm, and they have to be targeted in these inclusions in order for the cell to be protected during the stress response. And when it goes away, these occlusions, they get eliminated. It turns out that that also the protein quality control system, the proteasome and the ubiquity system is known to be involved in sort of dealing with the elimination of these condensates, mainly by degrading the misfolded proteins that are accumulating into there. Now, the interesting thing, the pathological interesting thing about the formation of these stress granules is that they're related to pathology to this new disease called amyotrophic lateral sclerosis. Because what's happening to this disease is that many proteins that they go into the stress granules, they are mutated in this disease. And in this case, instead of these condensates to become reversible, in other ways that the system can sort of eliminate it, these aggregates now, these condensates, they getting transformed into irreversible toxic aggregates that are accumulating in the motor neurons of these patients. And this accumulation of these aggregates, they are related to this motor neuron disease. So this causes a sort of neurodegeneration by sort of making toxicity to these motor neurons, and the thing about this disease is that is a fatal disease, quite rare at the moment from what we know, but the life expectancy is 2 to 5 years upon post-diagnosis, because upon degeneration of the motor neurons, the patients get muscle paralysis, and of course they die because of asphyxia. They cannot eat. It's a really horrible disease. And what is also terrible is that at the moment there's no treatment for this disease. So of course, there's a very big interest to try to define mechanisms of how to exploit mechanisms in order to devise therapeutic interventions for this disease. And Stephen Hawking was one of the famous patients that suffered and died of this disease. Now, it turns out that this enzyme that I just described you, this NETP1, is related to the elimination of both the physiological and the pathological sort of aggregates that I just mentioned to you. Here we have an assay where we can monitor how the stress granules are eliminated during the recovery process that I just mentioned to you. So here you see the granules that are formed upon stress in these green dots, and if you put cells during the recovery, eventually they get sort of eliminated by the protein quality control system. If we inhibit the enzyme, if we make a knockout of this enzyme, in other words, conditions where we have a lot of NADH in the cells, this somehow causes the acceleration of the elimination of the granules. In other words, the cell now is able to eliminate these granules much faster compared to the control cells. And we think that this is related to the biology of these granules, of how they make, because what we found is that both the number and the size of these granules is much smaller compared to the control situation. And this phenotype will come important later on in the discussion. Now, this phenotype of this enzyme is not specific for physiological granules, but also for the pathological ones. So here what we did is we got fibroblasts from patients which they suffer from the disease, and we asked the question how the inhibition of this enzyme can deal with the pathological now aggregates that are found in this patient. And here we performed the same sort of assay. So in this assay here, what you see is we monitor how fast this either the wild type fibroblast or the pathological sort of fibroblast can eliminate sort of aggregates or stress granules from the cells during the recovery period. And we did this in controlled conditions or when we block the activity of our enzyme. And in this case, to block the activity of this enzyme, we used some inhibitors that we developed in the lab. These are nanobodies that they are very potent inhibitors of this enzyme. And you basically introduce these nanobodies into the cells, and you can block the activity of this enzyme, evident here by the accumulation of NADH that is known to happen when the enzyme gets blocked. Now, over here, you see what happens in normal conditions. So here you see that the normal cells, they eliminate in their inclusions, 50% of the inclusions are eliminated within 1 hour of recovery. And that's the normal sort of recovery response that you see in this type of assays. Now, the fibroblasts that come from the patients, they're completely resistant. So this delta over here shows this lack of elimination of these aggregates that recapitulates what you see in the disease. Naturally, in the disease, the patients, they cannot eliminate these aggregates, and that's because of toxicity. Now, if we block the activity of the enzyme, we see that in the wild type situations, we see indeed that we can get the sort of the elimination, but critically, we see also that the pathological aggregates, they also get eliminated. So in other words, now the inhibition of this enzyme can cause the elimination both of physiological and pathological sort of inclusions. And this is related to the sort of survival of the motor neurons that are exposed to stress. In other words, that if we block the enzyme, this elimination of the aggregates is related to the improvement of the survival of this fibroblast during these experimental conditions. Now, the question that we want to understand is the mechanism, how potentially this enzyme can cause elimination of this aggregate. So in the lab, we do a lot of proteomics. So the idea here is to find which substrates are modified by NADH, and most particularly to find the site of the modification, exactly where NADH it goes on the substrate. That's a very important information if you want to extrapolate the biological outcome of this particular modification. Now, I'm not going to bore you with all the mechanistic details. We found many substrates that are potentially controlled by this enzyme, but the enzyme that eventually we end up working on is this enzyme called PARP1, or polydipyribose polymerase 1. This enzyme, it was very interesting and peculiar because it was known to be involved in the DNA damage response in the nucleus. Because upon DNA damage, this enzyme in the nucleus produces this sugar, which is called polydipyribose, which helps the DNA damage repair pathway. But this sugar that is produced upon this enzyme is also important for the formation of the granules in the cytoplasm, because what the sugar it does, it modifies many stress granule proteins are modified by the sugar, and it helps the sort of interaction with many other proteins in order to form these inclusions. So the production of this sugar upon PARP1 is absolutely critical for the formation of these stress granules. But the activity of this enzyme has to be very tightly controlled during the stress response. Because if PARP1 gets overactivated and it produces too much PAR, then these granules, they go from a normal state into a pathological state, into a solid aggregate. And indeed, PARP1 overactivation has been observed in many neurodegenerative diseases, and we think that this overactivation is part of the reason why you have the formation of bad aggregates, of this overproduction of the PAR. Now, our evidence showed that when we block the activity of this enzyme, indeed, this PARP1 was a key sort of mechanism by which NETP1 controls this stress granule formation. Because we found that in NETP1 knockout cells, the activation of PARP1, and here you monitor how much PAR is produced upon stress, is dramatically reduced in the NETP1 knockout conditions. Indeed, providing the evidence that the NETP1 enzyme really controls the activation of PARP1 during the oxidative response, and we thought that this lack of production of PAR, it could be the reason why potentially the stress granules are eliminated much faster compared to the control conditions. So as I mentioned, the key advantage here, when we do the mutation, the proteomic analysis, you can find exactly where NADH in this case goes onto your substrate, in this case PARP1. And what you can do, you can go make all these mutations, make a mutant of PARP1 that cannot be NADH-deleted, and ask the question, how my biological process now is affected. Again, I'm not going to bore you with all the details, but by using these mutants of PARP1, we found that we can rescue all the phenotypes we see in the NETP1 knockout conditions. In other words, now the granules, instead of being small, now they're becoming very big, and they are very difficult to be eliminated. Basically, we reverse all the phenotypes. And that sort of proposed the following model, mechanistic model that we're working on. So it looks out that the mechanism by which this enzyme controls this granules and aggregate formation is through the activity of this enzyme PARP1. This enzyme constantly needs to remove NADH from PARP1 in order to be activated and produce this sugar that helps the growth or the formation of these stress granules. If now we block NETP1, we have the hyperinodylation of PARP1. This somehow blocks the activity of this enzyme. You don't have the production of PAR. The granules are smaller, and now they can be eliminated much faster compared to the control situation. So that's very interesting. But again, we want to provide a bit of the proof of principle of targeting this enzyme for therapeutic intervention. So the model system we use in the lab is the small nematode called C. elegans. So what is interesting here is that you can do two very interesting assays. The first one, you can monitor in the brain of the animal how you make in granules. So here you see the stress granules made in the animal. But at the same time, you can see how the animals move, because ALS is a motor neuron disease related to the muscle biology or function, in this case of the animal. So this is what happens if you introduce an ALS mutation from the human ALS mutation in C. elegans. The first thing is that you develop really bad aggregates, really bad inclusions, and the animals now stop moving. So you have a very good correlation because between forming aggregates and stopping the mobility of the animals, which recapitulates what you see actually in the patients. What was very gratifying for us is that when you inhibiting the enzyme NETP1, so this is ULP3 in worms, you see two things. The first thing you see that the aggregates go away, the size that's much smaller, but critically, the mobility of the animals now is rescued compared to the pathological ones. So in other words, now we have the proof of principle that targeting this enzyme through the mechanism I just described to you could be potentially a therapeutic mechanism through the elimination of these bad aggregates. What is also interesting is that by deleting this enzyme in the wild type background is not toxic. So the animals can live without this enzyme, no problem. So that's also, for therapeutic purposes, is also very important. So this is, if you like, the summary of this part, which links the NEDOID pathway, this enzyme, to neurogeneration. We think that by eliminating these ALS aggregates that they are found in this disease, by inhibiting of this enzyme, we can potentially have a proof of principle for targeting this enzyme for therapeutic intervention. Now, for you that you may have captured, the mechanism that we sort of propose is that NETP1 is working through PARP1 inhibition. Many of you know that there are PARP1 inhibitors in the clinic at the moment. We can discuss later on why targeting NETP1 it would be more beneficial rather than targeting PARP1 itself in ALS. But this could be potentially a matter of discussion. Now, the second part now will move a bit about the ubiquity system, but more about cancer and what we know. And this is more sort of at the preliminary stage of what we've done. So you probably very well know, here in this audience, that inhibiting the proteasome has been a very sort of potent mechanism or therapeutic target, basically mainly for leukemias. And one of the mechanisms by which this inhibitor it has a beneficial effect is that it's inducing, protein aggregation. By blocking degradation of proteins, one of the things that these proteasome inhibitors do, they induce in protein aggregation. And you will know better than me in this audience here that these compounds have been approved for patients with myeloma. They're not very potent inhibitors, treatments for solid, but for multiple myeloma, they seem to have a very, very sort of potent efficacy. So now the reason why potentially this protein aggregation may be related to cancer treatment is that it turns out that tumor cells, they seem to be much more sensitive to protein aggregation compared to wild type cells. So in this experiment, it's a very simple experiment where what you do in this experiment, whatever here is you take the extracts either from normal cells or from tumor cells, and you ask the basic question: what is the potential of each cell extract to form aggregates between control cells and tumor cells? And what you can see here in this graph over here is that tumor cells, they are much more sensitive to protein aggregation compared to healthy cells. In other words, that if you induce protein aggregation in the system, tumor cells, they form much faster aggregates compared to the wild type cells. And in many ways, that's what the proteasome inhibitors exploit as a therapeutic approach. They exploit the ability of tumor cells to make aggregates much potently compared to the wild type cells. And that's why we believe, at least one of the mechanisms by which proteasome inhibitors can induce toxicity, preferential toxicity of tumor cells compared to normal, healthy cells. So one of the sort of examples of protein aggregation that we looked and we're working on that relate to cancer is in the nucleus, and this is related to a compartment which we call the nucleolus. So the nucleolus is a site within the nucleus that many of you may know that is inducing the production of ribosomes. So ribosomes is a machinery that makes protein synthesis, and all this ribosome biogenesis, it happens within this sort of sites within the nucleus, which is 11 nucleolus. So nucleolus is a fascinated compartment with the nucleus because it's very dense, as you can see here, and it's very, very difficult to do sort of immunostaining analysis. Now, the nucleolus was sort of detected or discovered, back in the 1700s, but even from the 1900s, it started to be sort of related to cancer because the size of the nucleolus is there's a very good correlation between the size of the nucleolus and tumors. For some reason, and now we start understanding, is that tumor cells, they have much bigger nucleoli compared to the healthy sort of cells. And indeed, the size of nucleolus is now being used for pathologists to detect cancer cells. In sort of more recent studies, many sort of diseases, which we call now ribosomopathies, they're related to the nucleolus because these ribosomopathies, which they are mainly related to anemia predisposition, they are found that they are related to heterozygous mutations in many ribosomal proteins. And ribosomal proteins is the proteins that they are related to the production of ribosomes within the nucleolus. So all these mutations, they now predispose patients to many of the anemias. So that's, if you like, another evidence of how this compartment, this nucleolus, can be potentially related to sort of development of cancer. In our case, what we found is that the nucleolus is maybe related to cancer development through the formation of protein aggregation. So here what we monitor how the nucleolus behaves upon prototoxic stress, conditions that they are related, that they're forming protein aggregation in all these conditions. As you can see here, that's the normal sort of physiological form of the nucleolus. And when you expose the cells to all these stress conditions, the nucleolus changes completely morphology, and one part of the nucleolus, it makes these aggregates, which they are sort of made mainly by ribosomal proteins. You can see the formation of the aggregates in this live imaging. So you can see here, this is a healthy nucleoli. You expose the cells to stress, and you can see that it will lose completely its reorganization, and you can start forming these nucleolar-related aggregates. So this is what is known and what is formed upon stress. But what was interesting for us is that the ubiquity system is very sensitive to this formation of the aggregates. So ubiquity and also this hybrid chains that I mentioned to you, this hybrid chains between ubiquitin and date, they are very specifically decorating these aggregates that are forming into the nucleolus. And we now try to understand what is the role of this signal in order to deal with this aggregate formation. We now believe that the formation or this accumulation of ubiquitin in these aggregates is related to helping how these aggregates will be eliminated during the recovery process. So here you will see that this, how the system recovers during the sort of recovery period. So here you start with an aggregate state, you put the cells to recovery, and then you will see that the nucleolus sort of recovers back into the normal situation. Basically has eliminated all these sort of inclusions. And in order to understand how the system manages to eliminate these aggregates, these nuclear aggregates, we do a lot of proteomics. So again, without giving you all the details, we're able to isolate these aggregates from the cells. We can interrogate by quantitative proteomics how proteins go into these aggregates during the formation and how they get eliminated. And also by the proteomics, we can identify the factors that are coming into the aggregates that are helping into sort of their elimination process. So one of the elements, one of the key machines that we found by proteomics that is coming very fastly into these aggregates is the nuclear proteasome itself. So again, by using assays where we can monitor how nuclear proteasome behaves upon stress conditions, we can see here by stainings that during the recovery period, the proteasome is coming where the aggregates are, and they are helping the proteasome is helping in order to eliminate these aggregates during the recovery period. And the signal for this is this hybrid chains that are found to be accumulating into these aggregates upon stress. So here, that's how the stress response works here. You expose the cells to stress. The ubiquitous system, together with NED, goes directly into these aggregates. You recognize them. The proteasome sees this change over here, is recruited to the aggregates, and it helps the elimination during the recovery period. Now, the additional interesting aspect in this is it was not just that only proteins or the proteasome is coming to deal with this aggregation, but also you need the production of RNAs to help the elimination process. So the nucleolus is a very particular environment because it produces a lot of RNA in homeostatic, but also upon stress conditions. And what we found when we interrogate these aggregates by RNA-seq analysis, we found that there's a whole bunch of non-coding RNAs that they're only expressed upon stress conditions. So here you see the expression of these non-coding RNAs in control cells. So basically there's no expression. You expose the cells to prototoxic stress, you form the aggregates, and now the system is exploding by the formation of these non-coding RNAs. I can tell you now that all the induction of these non-coding RNAs, they are required for the elimination of the aggregates during the elimination process. So in other words, here you have the cooperation of the production of RNAs with the ubiquitin system that it helps the elimination of these aggregates during the recovery process. We still don't know how, and that's something that we're working on. So one of the interesting things just to show you is that these non-coding RNAs, upon they get induced, they go directly, they're accumulating into these aggregates that they induce upon stress. And now you can see, so this is the nucleolus in controlled conditions. You expose the cells to stress, you're making the aggregates, and here by fish analysis, you're looking how this non-coding RNAs, one of these non-coding RNAs is localized, and you see that goes directly into these aggregates. And this aggregate accumulation, it will help eventually the elimination of the aggregates during the recovery process in a mechanism that we still don't know of. So here, this is a schematic of the process of how the system deals with this type of aggregates that are taken out in the nucleus. So upon stress, the system recognizes these aggregates. It will provide a whole bunch of enzymes that, like this enzyme over here, which will recruit it to these aggregates. It will decorate the aggregates with these hybrid chains. The proteasome will be recruited to eliminate, and still in an unknown way, in cooperation with the non-coding RNAs that are found here, it will help the elimination process. Now, one of the interesting things the things that we try to understand is that this nucleolar aggregate formation, how is related to cancer, how is related to patients that are dealing with this anemia process, and by potentially controlling this aggregation process, if we can have a beneficial effect for these patients. In particular now, what we're interested to see is the induction of these non-coding RNAs, if it can be used as a biomarker for these anemia diseases, because these non-coding RNAs are completely unknown, and we now think that this potentially expression could be used as a potential biomarker. So in the next 10 minutes, I will finish with the last project that is more new and also related to cancer development, and which is also related back to our enzyme, the NETP1 enzyme, because now I talked a lot about the bortezomib, which has been very successful as a treatment for leukemia, but this success prompts pharmaceutical companies now to target every single component of the ubiquitin system. So now the ubiquitin enzymes, the NED18 enzymes, the SUMO enzymes, they'll all be targeted for therapeutic intervention, because now we believe that these pathways, somehow they are related or they're implicated to cancer development. So I'm sure that you will know from Carlos' work that now there's this very highly specific inhibitors for the NED8 pathway that they block the E1 enzyme for NED8, the first enzyme of the cascade, which is called pevonestidat. And this compound now is in phase 3 clinical trials, mainly with combination therapies with other chemotherapies. And the main reason why the NED8 pathway has been targeted for therapeutic intervention is that it was known for many years that the NED8 pathway is upregulated in many cancers. And here I I sort of list a few, with the particular interest of ours in hepatocellular carcinoma. But still, why the NET pathway is upregulated in NET cancers, we still don't know, but we know that it's something potentially beneficial or potentially therapeutic target for cancer therapy, hence the use of this drug to block NEDylation as a potential chemotherapeutic. Now, just to give a few examples of what we've done in collaboration potentially with Malou Martinez-Santar here close in Bilbao, that she's a leader in liver pathogenesis, and she discovered that she was one of the pioneers to discover that the NEDylation pathway is upregulated in liver cancers. And here you can see that the studies that Malou and other people have done is that this upregulation of NEDate is sort of related with sort of patient life expectancy. In other words, that people with high levels of NET, they have lower sort of life expectancies, which is the classic sort of evidence that potentially the pathway is related to this disease. So NEDate is upregulated in cancers. We still don't know, how, but clearly the NEDate pathway seems, or inhibition of the NEDate pathway seems to be beneficial for liver treatment. And in this experiment, what you can see is that if you, in combination therapy with a sort of known and currently used sort of treatment for liver cancer, which is compound over here, which is called sorafenib, maybe. You can see here that the combination, it really improves the efficacy of the compound. So that's the sort of evidence that people have that shows that potentially targeting the NEDate pathway could be beneficial for liver cancer treatment. Now the question is how the pathway is upregulated. What is the molecular basis? And again, we go back to the basics of how the NEDate pathway over here. I described to you the forward reaction. NETP1, which is the denadylating enzyme. And as I mentioned to you, you block the enzyme, you have this massive accumulation of NEDate in cells. It turns out that this sort of enzyme now is involved in the potential sort of liver, in the hepatocellular carcinoma development. Remember that I mentioned to you for the amyotrophic lateral sclerosis, the activity of this enzyme was upregulated. What we found with the collaboration with Malou lab is that in cancer, you have the opposite effect. This enzyme now is downregulated, and particularly in live, in the tumor models for liver, for hepatocellular carcinoma, that Malou's lab is using a lot, you can see that, and that what was known, that you can see that in mouse liver tumors, the NEDate system is upregulated, but you cannot see here that the levels of this enzyme, they're completely gone. Okay, so here we have this paradox or potentially molecular explanation that this downregulation of this enzyme could provide potentially the molecular basis of this upregulation of NEDate that was known for many years in cancer development. Fine. The question is, what does this downregulation does? How potentially is linked this downregulation of this enzyme? So one of the biological functions, biological properties that we found that is controlled by this enzyme related to cancer is that its inhibition, it completely blocks the induction of apoptosis upon DNA damage. So again, here we're using C. elegans as a model organism, which is great for doing a lot of DNA damage biology research. So here, what we've done is we expose C. elegans in these crazy amounts of radiation. So it sounds crazy to you, but for C. elegans research, that's the normal radiation you use. And when you induce this radiation to this animal, you induce apoptosis, evident by the induction of this apoptotic copsy that you see in the germline of the animal. So that's the induction of apoptosis in these wild-type animals. And when you go back and you look in the NETP1 knockout cells, you see that this apoptosis is completely blocked. So now we know that by blocking this enzyme somehow it prevents the induction of apoptosis upon DNA damage. And this somehow seems to be related to chemotherapy and to potentially to liver hepatocellular carcinoma. Because what we've done here is we took the liver mouse tumors, and what we've done is we put back the missing component. We put back the enzyme, the NETP1 enzyme, and we asked the questions how these liver tumors will respond to chemotherapy, in particular to the induction of apoptosis upon the chemotherapeutic response. So when we put back the missing component NETP1, the first thing we do is that we restore the levels of NET in cells. In other words, so from this upregulation that you normally see in cancers, we put this enzyme and now the NET levels go down as expected, but critically, we now restore the response of these tumors to chemotherapy. So the normal wild type tumors, they have this type of induction of apoptosis. We put the wild type enzyme, and now we restore or we increase the sensitivity of these tumors to induction of apoptosis. Induce, confirming the hypothesis that the reduction of NETP1 we see in this cancer, which caused accumulation of the NET, may be the mechanism by which these tumor cells are resistant, and by reintroducing back the missing components, we can restore the resistance of these tumors to chemotherapy. So this is where we are with this project. We believe that NETP1 potentially is acting as a potential tumor suppressor in this case. So for cancer treatment, it's not going to be a very good therapeutic approach, but what we're looking for now is that the enzymes that they're working against NETP1. So remember that this reaction is reversible. So NETP1, it takes out NED, but by definition, there will be enzymes that they will put NED on the substrates, and that's the enzymes now we're looking specifically in hepatocellular carcinoma, because we think that these enzymes could be potentially therapeutic targets for this disease. So I will finish with this slide over here. I think what I tried to do is to give you an overview of how potentially these ubiquity-like molecules can implicate it to formation of aggregates, how you can potentially exploit aggregate formation for the treatment of disease, neurodegeneration, cancer development, and particularly for the system that we work, we think that we found a rather sort of peculiar role for this enzyme NETP1 in diseases, because it could be a biomarker of contrast. In other words, we found that in neurodegeneration, the activity of this enzyme is upregulated. And in this case, in this neurodegenerative disease, ALS model system, by blocking NETP1 could be a good therapeutic approach. In contrast, though, we found that in liver cancers, the activity of this enzyme now is downregulated. In this case, it's not going to be a very good therapeutic target, but we have found potential alternatives to target the opposing enzymes for NETP1 that could be used potentially as a good therapeutic approaches. With this, I will stop. I will thank the team that, of course, has done all these works. Particularly, I want to thank Emeric, that is using a lot of the C. elegans model system in the lab, together with Yola. Leo is doing a lot of the stuff on the protein aggregation, project. Rohit over here, he did most of the work together with Tufik on the neurodegeneration in NETP1. We have a lot of collaborations. Malu, which is very close here in Bilbao, she's been great. He helped us a lot with the development of project in hepatocellular carcinoma. We're still working with her quite a lot. Many collaborators that worked, with us, especially the proteomic facility in Montpellier. The funding bodies, thank you for listening. Thank you for coming and for listening.